the automated image annotation with ontologies using deep learning networks. And this is a project that started as part of the Planteome on how to build tools and train and educate our users. For example, if you're looking at an Erenchyma in a root, or you're looking at a mesophyll cells in a leaf, or you're looking at different types of fruits and different types of leaf shapes and leaf sizes and leaf forms, how do we educate our students and our breeders and molecular biologists or bioinformaticians to understand what structure we are looking at? So the idea was we can go and grab everything from Google and show those images and tell them, but that was not the purpose. Purpose was, can we use those images? For example, specifically, we can say that, show me all the apple fruits that you're looking at this image. Build those libraries, actually annotate the sections of the images, that this is a fruit, this is a leaf, this is a stem, and then actually make sure that the next time, so somebody has to do the annotate those images and sections in the first round, but the second time the same image comes, can we use machine learning to identify the same structures? So many people use Google Lens, has just launched, and some other platforms for doing those kinds of things to show me, can you identify the species, and you take a picture and it will tell you which species it is based on the characters it finds. So we were trying to build something for plant community on that sense. So we started with a software called AISO. It was for augmented image segmentation and annotation. So as you can see, let me have my pointer. Okay, so you can see in that this was a desktop application where you can actually run it and pull in an image, select the foreground and the background, and then ask it to find a segment. Once you find the segment, then the users were allowed to actually annotate those sections, whether this is a leaflet or a section of a leaflet and things like that. And then once those images are annotated, we can annotate with ontologies. Then you go to the ontology browser, and we should be able to show you that particular structure just to educate and train, right? So that was the criteria that we were looking at. But pretty soon we realized that everybody's cell phones were actually cameras were getting better and better. And the image sizes were increasing, and you could not handle, the software was not able to handle all those image sizes and the granularity and the resolution and things like that. Plus, the storage was a bigger problem. And the other thing was, if the person doesn't share the images, there is no way to build the machine learning algorithms for that. So we started working with Cyverse. I don't know if you've heard about it or not. It's similar, it is a new name, don't worry about it. It's a new name for iPlant Collaborative, and it provides you the virtual machines that we are looking at. So we started porting this tool on the Cyverse platform, where you can go and have high resolution images available. You take all the other tools, they have a lot of other BisQue tools that can be used for image analysis, and we added the segmentation and ontology plugins for that tool. So you can import the images, take out the background and select the foreground, and it will give you the segments that you should be looking at. So then we can annotate those images using the ontologies, right? So that's what was happening. But again, once those were available, we had two options. Can we use the neural networks to create their own catalogs and have their own learning? Or you allow the segments to be formed based on what we have already curated, saying that this is the fruit. So you were using both the sections, so both the options. So if you don't segment, then you can just say, find the objects in this image, and it should automatically go and find the objects based on the machine learning and tell you that there is a fruit here. Unless you want to narrow down and create a segment around the apple, and then you would be able to say that this is a fruit, right? So this is what was happening. In this case, we made a segment, and then we allowed it to find what that structure is, and then it automatically comes back and returns and says it's a fruit with 91% confidence that it is, based on the machine learning technology that we had. But again, we wanted to build, say, this is what we are having, but can we automate it more in that sense? So we started moving towards more into the neural networks aspects. Can we automatically identify or differentiate between the background and highlight the foreground images from that perspective? So we started with MATLAB and other things, and we are quickly moving into the Python infrastructure, and that is allowing us to identify those things. So idea is any images that are coming in, and then you should be able to identify and differentiate all those things as a first pass, and then you go and ask the user to engage and confirm those things that are happening. Okay, so in the machine learning things, what you're looking at over here, these are not planktons, but these are the patterns. So let's say an image comes and it starts taking account of the pixels that are in the images, and then starts categorizing what is this pixel and how does the pixel of its neighborhood differentiates between them. So it's trying to find the patterns and categories inside them. So it creates all the networks and connectors of those pixels and how many of those are forming. So it creates catalogs of those pixels around it. And so in this case, for example, we used about 5,000 images to classify a flower, fruit, leaf, stem, and entire plant. Those were broadly annotated by image clef, and we used those to train our machine learning software. And then what happened was next time if a fruit comes in, then we should be able to identify. But so that was available, so accuracy came out to be about 95%. In this situation, we could identify. That's it. So the idea was not just to identify the object, move a step next. Can I identify the characters of those objects? So what is the fruit shape, size, color, any spottings coming up on that? Or if it's a leaf, are we finding what kind of lamina is there? What's the width and things like that? What kind of curvature is on the margins and things like that? So we built another desktop applications. We took all the images that were originally in the image clef collection and started annotating using the taxonomic qualifiers that the anatomists use all the time. And so once we have that library, our goal is to get about 1,200 images. We are close to around 500, 600 now. Once those are available, then we are able to identify the characters of each leaf. So these are not limited only to the things that I'm showing you. For example, if it was a disease, or as we were talking with Ted yesterday, that based on the oxidation or based on the sections of the potato or sweet potato that we are looking at and the coloring patterns, if we can build the libraries with that, the same algorithms would work on classifying for certain things that you're looking at. So this is, for example, an image comes in and you want to identify and what that is. It tells you that it's a serrated margin leaf that we are looking at, and the ontologies come in that sense. So the tool is now running online, and hopefully, where is my mouse? So you can see a quick one. You select an image on the BisQue platform, upload it, and select the parameters, ask it for segmentation and identifying. So it's identifying its foreground, selecting its background. You have an option. If you don't want to use that option, then it will do it automatically. Create the segment quality that you want to create and run an algorithm, and it will automatically tells you the first pass of the annotations that would show up, that it's a leaf and it has this kind of a shape and size, and it's a serrated margin and it's a complete margin, and it's a simple leaf and not a compound leaf that you're looking at. So this is working on this kind of data sets, but we will start working on it as we move along in that sense. So that opens up more collaborative effort. If there is an interest in the community, we would love to work with you guys on creating some of these things. All the ontologies are coming from the Planteome and the crop ontologies, RESTful APIs that are loaded into the system. And it gives you an option, basically, to use the ontology trees on over here to find out from a collection and characterization saying, show me all the leaves that have these kinds of curvature, or show me all the mutants that have these kinds of properties using the tree. But the images are already annotated and the segments are already annotated. So if there are disease images, there are pathogenic images, there are fruit plant conditions images, we can use those to build different kinds of libraries, and you should be able to annotate those kinds of things. That's the next round of Planteome we are thinking about. This thing will be running on the Cyverse data store and available to the community from that perspective. So it's been a big project. There's a team from the Cyverse, Neerav Marchant, and B.S. Manjunath's group in UC Santa Barbara. And much of the work was done by Dimitri Trigakis, and is a PhD student in Oregon State in computer science, and Justin Priest from my lab, and all the Austin Meyer and Justin Elser in my lab that have been working. And the work is being guided by Sinisa Todorovic from the computer science department at that place. So thank you.